Welcome everyone to our new episode of CXO Talks at PharmaShots. Today we have Vikas Sharma with us of QurAlis. He is a Chief Business Officer. So before we start, a quick intro from my side. Hi, I'm your host Aditya Singh. I'm father of two boys and husband of a superwoman. By education, I'm a pharmacist and I love to travel and explore new things. Today, we have a special guest, uh, Vikas Sharma, who is a Chief Business Officer of QurAlis. And if I talk about QurAlis is a company which is working deeply into the neuroscience and helping the patient in neurodegenerative diseases. So, Vikas, over to you. If you give us a brief intro about yourself and your journey so far, and then if you can talk about the QurAlis.
Thank you, Aditya, and thank you for inviting me to this podcast. I'm excited to be here. So, as you mentioned, I'm the Chief Business Officer at QurAlis, which is a company based in Cambridge, Massachusetts. I personally, I've been at QurAlis 4 years now. I'm an immunologist by training, but always been on the business side of large pharma like AstraZeneca, MedImmune, mid-size like Macrogenics and BioXcel Therapeutics and others. I love science, and what I say to people is like I like to do business with the science, right? So that's what that's what my passion is, and bringing therapies to patients, and especially for diseases that are unmet need is part of that passion. QurAlis, as you described, we are a precision neuro company based in Cambridge. We are our first foray is in ALS, a neurodegenerative disease, but we are expanding into other neurodegenerative and neurodevelopmental diseases like FTD and Fragile X. We have also a program in neurology now, in epilepsy, and I can tell you a little bit more when we talk about our programs as we go forward.
Would you give us a brief background how this company started, and then what was the idea or what was the need which the founder looked into, and then they started the company? I can see they were the Harvard med school graduates, and then they started this company.
Yeah, and the company started in Harvard, as you are absolutely correct, and it was a need that's in the field that is required. So neurodegenerative diseases, these diseases there's no animal models for these diseases, so it's very difficult to study them. And when you can't study them in animals, it's very difficult to also discover targets or targets that cause these diseases, right? So this was the big discovery invention at Harvard at Kevin Eggen's lab, right? He was the first person in the world to make motor neurons from stem cells. And that changed the field, because what they did was made models of how you can test the drug, test the target, and to study these. You know, it's not difficult to go after or make therapeutics if you know exactly what you want to go after, right? The biggest problems in these diseases are what is causing the disease. And I think by discovering and inventing these models, it was easier to study. So that's why our first couple of targets that we are going after were discovered in Kevin Eggen's lab, right? So these are the genetic drivers of the disease in ALS, but their use may be broader than ALS itself, because just like what happens in oncology when you have these targets that are relevant to multiple tumors, similarly in neurodegenerative diseases, these targets may be relevant for not only ALS, but now we know that it may be relevant for 50% of FTD and 30% of Alzheimer's. That's how the company got started, right? And we started as a therapeutics company. We didn't have a platform when we started, but the need, because we were going after targets, and so whatever modality fit that, we would do that. That's why we have ASOs. We are developing ASOs, and we are also developing small molecules. Now, when we were developing the ASOs, we found limitations of these ASOs. So we then discovered a new platform or invented a new platform for ASOs as well, which we can also talk about in more detail. But that's how the company started, and that's what was missing in the field. And now everybody's doing this basically. When the company started, when Kevin Eggen came up with the models, it was not that prevalent. Now, wherever you talk about people doing neurodegenerative diseases, everybody works with these STEM models, right? They make different kind of motor neurons from, cortical neurons and others. And that has changed the game. And that has changed how companies approach, discover new targets and approach therapeutics.
Yeah, as you mentioned about the proprietary your proprietary technology or proprietary platform, FlexASO. So could you tell us about that and why it's gaining a lot of attention in the neurodegenerative space
Yeah,
or
so
like in the therapeutic space?
Yeah, definitely. As you know, ASOs, antisense oligonucleotides, have been around as therapeutics, right? People have been working with this. People have been developing this for multiple decades. In fact, one of the most successful products of ASO is Spinraza, that is marketed by Biogen for SMA, right? It's a $2 billion drug. But there are two kinds of ASOs. There are ASOs that are called gapmers, and then there are ASOs that are splice modulation or RNA restoration. So in gapmers inhibit RNAs, and splice modulator upregulate RNA or upregulate protein. So there are two distinct functions depending on how you make these ASOs and how they are designed. Okay, so our expertise and our platform is dedicated to splice modulation or RNA restoration. This is where we play. This is our therapeutic. All our therapeutics are also geared in that direction. The problem in these with ASOs were that not now, earlier, was that there was a class toxicity, because if you like with any therapeutics, right, it takes time to refine them, takes time to make them better. And if you put too much of ASOs, because ASOs have a very long half-life, if you give too much of the ASOs itself accumulates and can give class toxicity. They also can have off-target activity and other aspects. So what we try to design is ASOs that would have longer half-life, would have less off-target activity, would have more potency and a better biodistribution. So biodistribution was another important aspect. So when you give ASOs, either through IT administration or you give IV administration, right? In IT, you're putting straight into the spinal cord. So you are relying on the normal distribution of fluids for the ASOs to distribute in the brain. But if you want ASOs to go to different parts of the brain, for example, to the cortex, to the deep brain areas, they may not go because of poor distribution. Now, the first disease we are going after is ALS, which is mostly motor neuron disease. So it's all over the spinal cord, right? So it's easy. That's the area where ASOs can distribute very easily. But we wanted to, because as I mentioned, we are going beyond ALS as well in FTD, which is cortical disease, and we want these ASOs to distribute better. So in all our studies, so we ground up, redesigned the whole architecture of ASOs. So it's not using the same backbone architecture that traditional ASOs use, but we changed the backbone itself, and that's what was novel. And one of the other very surprising feature that we found was because of this that there is a reason we call it flex, because it offers the flexibility. And because of that feature, we are able to go to regions of the RNA that traditional ASOs can't. And what it offers is, besides regulating RNA or correcting splicing, we can actually modulate protein expression as well. Modulation, what I mean is when you don't want 100% upregulation, you only want 50%, you can do that. We can do that with our FlexASO platform. Traditional ASOs can't.
Mm-hmm.
So if you take all these 4 or 5 features, that differentiates our platform from what is available currently, that's what distinguishes us, and that's what is attracting a lot of the pharmas. I mean, we did a deal last year with Lilly, as you may have heard, that involved our FlexASO platform, and we are in
Mm-hmm.
discussion with multiple companies right now as well on these Flex collaborations.
Yep. So you already answered my next question, which is like, are you going to looking for any strategic collaboration or for future or for current indication? So you already answered that. Let's jump into the pipeline
But
candidate.
I can expand a little bit more on that. I would like to,
Yeah,
which is
definitely. Yeah.
yes, that's through inbound interest that we are having this conversation. But
Mm-hmm.
because what Flex has platform has also offered us is to go into other diseases, right? So to that end, when we find targets in other diseases like Fragile X, for example, and we'll talk
Yep.
about the UMass collaboration where some for a unique discovery by UMass, but where RNA restoration or splice modulation correction was needed, we can
Mm-hmm.
utilize our platform to do that very effectively.
Yep.
Similarly, we have a program in PSP as well where we are rebalancing. And we have multiple other programs in the background that are not made public yet, but we are working on. So but as a company, we are not that big, right? So we can only work on so many programs at the same time, and
Yeah.
that's one of the strategic reasons that we also want to work with other companies and also expand. If our platform works, why not
Yeah.
utilize for other diseases that we are not working on? And that's the idea, and that's what we are trying to do.
And obviously, if you do a partnership or you do a collaboration there, it increases the chances of getting other indications to be leveraged, and then other therapies can be built easily in that way. So it's a win-win for both of the companies, yeah, if I would think in my perspective.
Yeah, definitely.
All right, so let's discuss about the pipeline candidates. The lead candidate QRL-201 and QRL-101. So can you little like talk about these product little bit so that we can understand how it's going, and then uh, what is upcoming for the neurodegenerative diseases.
Definitely,
Yeah.
definitely. I'll talk about our first program, QRL-201 first, which
Yeah.
is a statin 2 candidate that corrects the missplicing of statin 2.
Mm-hmm.
Now, TDP-43 has been now attributed as a hallmark of these diseases. TDP-43 is an important transcription factor, right, that binds RNA for its proper processing. In a lot of these neurodegenerative diseases like ALS, like FTD, TDP-43 is missing. It comes out of the nucleus into the cytoplasm. So then the RNA or statin cannot be processed properly. What we do with our molecule 201 is it goes and binds to the splicing site, right, and corrects the missplicing. And so then the RNA is able to be processed properly, and the protein is being produced. How we found this out was by in Kevin Egan's lab, right, and looking at autopsy data, autopsy, that 90% of ALS patients, sporadic patients, where you don't know what is causing the disease, this protein is missing. So logically, if you think about an important protein, statin, which is crucial for neuronal health, microtubule formation, transmission, vesicle transport. If this protein is missing, it can only cause problems, right? And if I'm able to replace this protein through this sophisticated therapeutic, then
Yeah.
anybody with sound thought process would think that it could only offer benefit. That was
Yeah.
our idea, and that's our idea. But very importantly, we don't want to rely completely on clinical outcomes. So that's where the precision part comes in. we have biomarker associated with all our therapeutics. So biomarkers for target engagement, biomarkers for mechanism of action, and biomarkers for efficacy. What we want to understand is when you give a therapeutic, right, are you actually engaging the target? Is the therapeutic hitting the target? That's the first thing for us to know, because otherwise you would be looking for things downstream, whereas you don't even know your therapy is hitting the target. Then if it does hit the target, is it causing the downstream effect? And that's the mechanism of action, right? You can look at PD markers to see whether you're having an effect. Because remember, the clinical effect is the last part of it, right? If you're not able to cause the biochemical changes that would result in the clinical effect, but you don't want to wait that long. You can see things early on with these biomarkers. And that has been our thesis, and that's how we are progressing these therapeutics. So 201, which is in the clinic, we have finished our enrollment in phase 2 POC studies. We'll be announcing that data next year, basically. So that's very exciting. It's the most anticipated target in this space. It's a disease-modifying target. If you may have seen the news of tofersen, that was approved for SOD1 therapeutics for ALS by Biogen. That is the first ASO, right, that actually causes disease modification. And it's only 2% of the total ALS population, right? But it's a great proof of concept, right, that came about. Now it's on the market. And what I heard in different conferences from lead PIs is that now that a lot of patients have been on the drug for 2 years, 50% of patients are regaining function back. That has never happened in ALS before. That is super exciting. So what that means is that when you actually cause disease modification, especially with a genetic target, right, you may not see the immediate effect right away, but as the patient as the drug stays on board and as the effect becomes more pronounced, and you're able to arrest the disease, that you will see the effect later on, right? That's why we follow these patients for long. So that's very exciting target that we are going after. The second molecule is QRL-101, which is our small molecule program. That's the only small molecule that we have in our pipeline. It's a highly specific Kv7.23 agonist. Now, Kv7s have been known for a while. There was actually a drug on the market called retigabine, in epilepsy. There was also a drug in Europe, flupirtine, that was a Kv7 agonist for pain. But these were not specific drugs, like they were pan agonist, so they had a lot of liabilities. And so these drugs were withdrawn from the market because of the liabilities. But some of our founders had done clinical trials when the drug was on the market in ALS patients, and they found that there was a biomarker effect with hyperexcitation. So hyperexcitation is also a hallmark of a lot of diseases, and so was in ALS. So there was an effect on the biomarker. So then we decided to make, and we worked with Eli Lilly. Actually, we licensed this molecule from Lilly, and we worked with them and to come up with a de novo 7.23 agonist that is highly specific. It does not bind any other subtype 4, 5, or even GABA. And that gives us the confidence that we know that the target works. We just need to remove the liability from the molecule. But other companies like Xenon and Biohaven also developing these targets for epilepsy, which is already a validated target actually, and other behavioral diseases. So we are also advancing this program into rare epilepsies and potentially pain as well. So we are exploring those options, and stay tuned for the news. We'll be initiating proof of concept studies in multiple diseases in the near term.
I will definitely wait for the news to come. And do you want to discuss anything else apart from these two lead candidates? like what else you are like, what other indications you are working upon?
Yes. So as I mentioned, ALS, I covered ALS. We are also planning to go into epilepsy.
Mhm.
We are planning to also exploring frontal temporal dementia. So that's something because, as I mentioned, 50% of FTD patients also have TDP-43 pathology. So
Yeah.
potentially our statin molecule may work in that space as well. Even our
Hmm.
UNC13A target or molecule may work in that space as well. So we'll explore in the future those diseases as well. And then I mentioned that our program in Fragile X that we are going after. So that's a program that we in-licensed from UMass. The foundational IP from Joel Richter Lab, who discovered a brand new mechanism, right? Of and that makes the disease a monogenic. Basically, it's one missplicing that happens in about 70% of these patients. And if you're able to correct that missplicing, then you can have benefit in that disease. It comes with a biomarker, all the bells and whistles that are needed, right, to develop these programs. So
Yeah.
we are advancing them very rapidly towards candidate and clinic.
Perfect. All right, so it's getting a bit scientifically very heavy. I would like to lighten the mood little bit. So, would you like to tell about a little bit about yourself as a person and how you usually spend your leisure time, or what are your hobbies? So, our our listeners can, if they
Yes,
want to know you
between
more, yeah.
as you mentioned, you have two boys. I have two girls actually, and so obviously, I would like to spend a lot of time with them. They are still young, so I like to spend a lot of time with her. Besides, when they are busy and when things, I like to ride motorcycles. I'm also a very avid target shooter, so I was actually um, I do a lot of skeet trap shooting in my leisure time as well. I love gardening. So in the summers, I do gardening as well. I mean, this is one of my passion to actually build a greenhouse so I can enjoy fresh vegetables and salad all year round. So that's something that I'm gearing towards and trying to do that also
Mhm.
in the future, hopefully. And I'll definitely keep in mind to invite you for some fresh vegetable and salad when it's ready.
Absolutely, 100%. So, and I'm happy to know that you are still able to get time with your all of the busy schedule. Let's jump on to, you already hinted a little bit about the exclusive license agreement with UMass Chan Medical School. Can you put some light on that agreement?
Yes. So as I mentioned, like we developed this platform that can be effective in a lot of other diseases, right? And
Mhm.
again, multiple targets. So when we became aware of this unique discovery at UMass
Yeah,
um, about around Fragile X and a new target discovery, which fit our strategy and our therapeutic strategy pretty well,
yeah,
right? So we didn't then work for a year with them actually to replicate the data and confirm, right? And it was and once we did that, we then exclusively licensed the patents from UMass Joel Richter's lab, and we are also working with them in parallel. We're also working with other KOLs because as you know, it you'll have to keep the scientists and the clinicians, they have the most experience around these to work with you. And that has
yeah.
been our thesis as well in the past. So we always try to work with them and gain their insights and their expertise in this area. So yeah, we licensed that in, and now we are working on this program very actively. Um, and hopefully, there'll be a lot of updates in the future coming around. And one other piece I missed was They also had found biomarkers. They were developing biomarkers to identify this patient population, and we have access to the biomarkers as well. So we are also working on developing that biomarker in parallel, because very importantly, it's important to identify the right patients to get the therapy, right? And this is where the precision part comes in, right? And this is where the probability of success for the molecules come in. Has been the thesis of QurAlis, and this is honestly speaking, this is what the future of neurodegeneration is.
Awesome. So after this, I would like to dive into the future technology, which is artificial intelligence. And being a AI enthusiast, I would like to ask how this technology is shaping the future of healthcare, especially the neurodegenerative therapy area, and how you are utilizing AI and ML to get your drug development upgraded or taking to new heights or whatever way you want to call it. So, what are your perspective and thoughts on this?
Yes. So, innovation is in all aspects, right? When you're an innovative company, you utilize that from all aspects. Now, AI is not a new thing for us. People have been using AI. I mean, now it's more fancier to use that term, but I'm pretty sure you are aware that labs were utilizing these algorithms for a
Mm-hmm.
while now to do analysis and other things. I mean,
Yeah.
and companies have been utilizing these for discovering new target. It actually what it offers is efficiency, right? Above all, you're able to go through a lot of data in a short period of time, and you can build your algorithms around to get the answers that you want.
Mm-hmm.
Something that would take a week to analyze now takes a
Yeah.
few hours, and that creates the efficiency, and that makes us better. So we utilize machine learning algorithms throughout our process, right? From early discovery of new targets to where the computational aspect is used to actually developing or coming up with the hits uh, against the targets as well, right? Because you can utilize, because that's all initially done in silico. It's all in silico designing, and now you can
Mm-hmm.
utilize that as well in
Yeah.
doing so. Then in the labs, we have our biology lab where we have our own stem cell labs, and then we have our FlexASO labs, and we utilize these algorithms all the time for all different aspects, right, of analysis. But besides that, we also have a deal that you may have heard of. We did a deal with Unlearn. So besides in the research side, where target discovery, hit discovery, all those things that are utilized for analysis. Besides that, we are also collaborating with a company called Unlearn for at the clinical side. So as you
Mm-hmm.
know, these diseases are very tough, and nobody wants to be a placebo, for example. So Unlearn is a company, and I think there are a few other companies like that, but Unlearn was one of the pioneers there to make a twin RCT. So basically, they make a twin AI twin of the patient,
Yeah.
right, for trials. So just imagine that the patient is on the trial, and their placebo control is the AI twin, right? So this is, I mean, none of these have been approved yet or have been blessed by any of the regulatory agencies to actually
Mm-hmm.
run any official trials yet.
Yeah. Yeah.
But in order to pioneer in any of these areas, it's sort of like a validation. So you have to run the validation. We are running our trials with placebo, but we'll also in parallel compare that, okay, this is the placebo data. What does the AI data looks like, right? And if it's matching, then that gives more and more confirmation and
Yeah.
comfort to
Yeah.
agencies, to us, that okay, now you can utilize that.
Mm-hmm.
So that's what we are doing, and hopefully we can utilize that even more in the future as there's more and more um, acceptance of these new technologies are there.
Yeah.
We can, like, instead of having 30% placebos, you can have less placebos, and you can power the study better. That's another aspect that you can utilize. So we are pioneering not only in the first-in-class targets, we're also pioneering the way we run these trials, the way we analyze these trials, in all whatever it takes, right, to do the most effective study and bring these therapies in an efficient and an expedited manner to patients. Because remember, I mean, ALS patients only have 3 to 5 years to live. And if we spend 3 to 5 years running trials, then we will never have these drugs available for these patients. So we're whatever we can do to get these therapies that can be effective to them is we should all utilize.
I would say that's a great news. In your understanding, what is the usual reduction of the time? So when initially I was studying pharmacy, it was told that it is 16 years from the lead generation to the market. So what is your thought? Like how much time has been reduced with the use of these new technologies?
So there's still to come. A lot of these things are being utilized now, but you have to imagine that those numbers that are presented are usually is generic for all diseases. You cannot compare a migraine drug and an ALS drug with the same lens. Can look at that in the same lens because of the fact that the way that trials could be run. So you can run adaptive trials, for example, in these diseases that are highly unmet need. Remember also when you're doing ASOs, for example, there is no placebo, there is no healthy volunteers, right? It goes straight into patients. So you're already, although you're doing safety and tolerability, you're also getting data in these patients. So you can move along these trials very quickly, where you can adapt these trials. For example, a phase 2 can be adapted to become a phase 3. And in those situations, then the therapies can come to the market faster because there are avenues like priority review, breakthrough designation, orphan designation, all these can be availed. And the diseases that we are going after are in the rare category. So it can avail all these different regulatory luxuries, basically, right? That can bring these therapies faster to the market. So I would say our goal is to do this faster, right? And in a lot of cases, for example, when Amylyx had the data, their phase 2 data, right? The advocacy group and the agencies asked them to file for approval. They were able to get approval in right after phase 2. So we believe that if you have effective therapies, that the need is so grave that we will be asked to come to the market faster. So in that case, then if you look at those timelines don't apply, right? They are gray. It all depends, right? How fast. And in our case, when we are doing these things, right now we can get from scratch to the clinic in 3 to 4 years, right? And then if you're able to design effective, efficient trials, right, you can run these trials also in 3 to 4 years and get to market. So there's high probability and good chance that from scratch you can get these drugs less than 10 years into the market, right? Because the patients need it and everybody understands that, right? And also the risk profile, right, is more tolerable because if you have a terminal disease where a patient's going to die, the risk profile, right, the risk appetite for a drug is different compared to, as I said, like a migraine. Right? So all these different aspects you have to consider, and not to look at all the therapies with the same lens. What the timeline that you mentioned would be for something that is very general population, lot of safety needed. Because remember, running trials is about recruitment, and a lot of the time is spent trying to recruit patients. And this is where again, I'll come back to the AI unlearned collaboration, because the less number of patients you can have as a placebo, right, you can reduce the time of recruitment also, right? So all those things will play a role, right, in reducing the timeline. That's our goal. We want to do that. I think the agencies want to do that. I think the advocacy groups, the patient want us to do that. So I think
Obviously, yeah.
everybody wants, and there's a lot of support to find innovative ways to be able to do that.
So we are almost to the end of this podcast. I have only the last question that what do you see in near future for QurAlis, and what are the key milestones which you are excited about?
Yeah, so I always tell when I'm having a conversation with investors and the banks and and when I'm presenting that we just getting started, right? Because it's to be honest with you, I mean, as you develop drugs, you learn, right? And when you learn, you become better. For example, the first ASO when we did, it took longer, right? Because we were
Yeah.
learning as we were doing that. The second one didn't take that much time, and the third one definitely doesn't, because now we know what the pitfalls, what things to watch out for. So we keep getting better at what we are doing. And now that our programs are in the clinic and going towards, as I mentioned, 201 will have data coming out next year,
Mm-hmm.
hopefully in the first half of next year. And then you have 101 starting multiple trials as well. So there's a lot of things a lot of catalysts, right? A lot of news coming out of QurAlis in 2026. A lot of the other earlier program will also advance to the next stages. We'll have news from with the molecule that we licensed to Lilly as well, right? We because it's exclusively licensed to Lilly, they will have news around those molecules as well, hopefully as well. So there's a lot of good things happening. We may have also collaborations around our FlexASO platform,
Yeah.
and all those things are going to provide a nice um, news cycle for QurAlis and overall, if you look at right, neuroscience is very hot. The two largest acquisitions this year was Intracellular Therapeutics and yesterday Avidity Biosciences. They're both
Mm-hmm.
neuroscience companies. The IPO that went out today, MapLight, is a neuroscience company, right? So neuroscience is hot. Neuroscience and I think it's hot because of new targets, new insights into the diseases, that is fueling
Yeah.
this excitement, because it's almost like the next oncology, what was 20 years ago, is the next neuroscience is going to be, right? Is the next 20 years, and we are at the cusp of it.
Mm-hmm.
We are also leading this pack in,
Yeah.
especially in the
Yeah.
neurodegenerative space and these diseases. So we are very excited. We are super excited to be able to do this, and and uh, and also seeing that the markets and everybody's coming around, realizing and understanding that yes, this is where we all need to be. I also not to forget RNA therapeutics. Everybody's talking about RNA therapeutics, right? I mean, that's what
Yeah.
everybody is all about, right? So
Mm-hmm.
that's again because of the less attraction to gene therapy. RNA therapeutics has become, is what everybody wants to do now as well. So we are very excited, and we have the platform, we have the programs, we have going after the diseases that are hot. So we are really excited, and we hope to update you guys in the future as we start getting more and more data.
Right. I wish you all the best, and I would love to hear the stories when we meet in future. So how it's everything going, and thank you for giving your time today and telling the insights of the neurodegenerative diseases, and then also about the QurAlis, how QurAlis is working to help patient and to get the ALS therapeutics in the market. Thank you, Vikas. It was nice talking to you. For our listeners and people who are watching on our YouTube, I request you to please subscribe our YouTube channel. That's PharmaShots. and I'll see you again pretty soon. Thank you.